The history of a life on Earth is simultaneously a history of extinction, and understanding this dual nature requires examining not merely the dramatic disappearances that capture public imagination, but also the intricate mechanisms, ecological relationships, and environmental circumstances that determined which lineages persisted and which vanished from the planetary record. When we speak of extinct animals, we are addressing a phenomenon that has claimed an estimated 99% of all species that have ever existed, a staggering proportion that reveals extinction as the statistical norm rather than the exception in the deep history of biological diversity. This observation fundamentally reframes how we might approach the subject, moving away from treating each extinction as a tragedy, requiring explanation, and toward understanding the conditions under which species manage to persist at all, across geological timescales measured in millions and hundreds of millions of years. The fossil record, which provides our primary window into extinct animal life, presents both extraordinary opportunities and significant interpretive challenges that shape everything we can claim to know about vanished species. Fossilization itself is an exceptionally rare process, requiring specific combinations of environmental conditions, sediment chemistry, burial speed, and geological stability that occur in only a tiny fraction of the places and times where organisms die. Estimates suggest that fewer than 1 in 10,000 individual animals become fossils under typical circumstances, and this proportion drops dramatically for soft-bodied organisms, small species, and those living in environments hostile to preservation such as tropical rainforests or mountainous regions. The consequence of this preservation bias is that our knowledge of extinct animals systematically overrepresents large-bodied species, those with hard shells or bones, inhabitants of marine environments and floodplains, and lineages that existed during geological periods when conditions favoured fossilization. Understanding what we cannot know proves as important as cataloguing what we have discovered. The temporal distribution of fossil sites creates additional complications for reconstructing extinction patterns and understanding the ecological circumstances of vanished species. Certain geological periods are represented by abundant fossil localities spread across multiple continents, while others are known from only a handful of sites that may represent atypical environments or unusual preservation circumstances. The Cambrian period, beginning approximately 541 million years ago, illustrates this unevenness through a small number of exceptional preservation sites, most famously the Burgess Shale in British Columbia and the Chengjiang deposits in China, that provide remarkably detailed snapshots of early animal communities but leave vast geographical areas and temporal intervals essentially unknown. The animals preserved in these extraordinary localities, including creatures like Anomalocaris, a metre-long predator with compound eyes and grasping appendages, and Opabinia, with its 5 eyes and forward-projecting proboscis, reveal morphological diversity and ecological complexity that would be entirely invisible without these windows into soft tissue preservation. Anomalocaris deserves particular attention because it represents not only an extinct animal, but an extinct body plan, a fundamental organisational scheme for animal anatomy that has no living representatives and no descendants. During the Cambrian period, multiple such body plans coexisted, each representing a different evolutionary solution to the challenges of animal existence, and the eventual extinction of many of these lineages cannot be explained simply through competitive inferiority or environmental change, but rather reflects the contingent nature of evolutionary history in which chance events and historical accidents shaped which organisational types persisted. The radiodont body plan exemplified by Anomalocaris, featuring lateral swimming lobes, a circular mouth apparatus with tooth-like plates, and a particular arrangement of body segments, dominated marine predator guilds for tens of millions of years, before disappearing entirely by the end of the Devonian period, leaving no trace in subsequent ecosystems except as fossils and as evidence of possibilities unexplored by later evolution. The study of extinct animals necessarily involves grappling with the incompleteness of evidence and the provisional nature of reconstructions, a methodological reality that shapes both scientific practice and public understanding in complex ways. When paleontologists describe the appearance, behaviour or ecological role of an extinct species, They are constructing interpretations based on fragmentary physical evidence, comparative anatomy with living relatives if any exist, inferences from functional morphology, trace fossils such as footprints or burrows, and increasingly sophisticated computational and chemical analyses. These interpretations change as new evidence emerges and as analytical techniques improve, meaning that our understanding of even well-known extinct animals can shift substantially over relatively short periods of scientific time. The history of dinosaur science provides numerous examples of such reinterpretations, from the recognition that many theropod dinosaurs possessed feathers to revised posture reconstructions showing active, horizontal body positions rather than the kangaroo-like stances depicted in earlier work. The question of why animals go extinct admits no single answer, but rather encompasses multiple categories of causation that operate at different scales of time, space, and biological organization. At the most immediate level, extinctions result from population declines below viable levels, caused by factors including habitat loss, direct killing by predators or competitors, disease, inability to find mates, and genetic deterioration through inbreeding. These proximate causes, however, typically reflect deeper structural changes in environments, ecosystems, or climates that made previously viable populations suddenly vulnerable to extinction pressures that would have been inconsequential under earlier conditions. Understanding extinction therefore requires tracing causal chains across multiple levels of organization and multiple temporal scales, connecting long-term planetary processes to immediate population dynamics and individual organism survival. The 5 major mass extinction events recognized in the geological record demonstrate the range of ultimate causes that can trigger widespread species loss. The End-Ordovician extinction approximately 445 million years ago appears connected to glaciation, sea level changes, and oceanic chemistry shifts associated with the movement of the supercontinent Gondwana over the South Pole. The Late Devonian extinction, which occurred in multiple pulses between approximately 375 and 360 million years ago, may reflect volcanic activity, climate instability, and changes in oceanic oxygen levels associated with the spread of terrestrial plants and their effects on nutrient cycling. The End-Permian extinction, the most severe in Earth's history, eliminated perhaps 90 to 95% of marine species and significant proportions of terrestrial life, likely through cascading effects of massive volcanic eruptions in what is now Siberia that triggered ocean acidification, anoxia, and extreme climate warming. The End-Triassic extinction cleared ecological space that enabled dinosaur diversification, probably through mechanisms involving volcanic activity in the Central Atlantic Magmatic Province. The End-Cretaceous extinction 66 million years ago, caused by asteroid impact and possibly exacerbated by volcanic activity in the Deccan Traps, ended the era of non-avian dinosaurs while leaving many lineages surprisingly unaffected. Each mass extinction reveals distinctive patterns in which types of organisms survived and which perished, and these patterns provide insight into the factors that confer extinction resistance or vulnerability under different environmental circumstances. During the End-Permian crisis, organisms with high metabolic rates, limited ability to regulate body temperature, restricted geographical ranges, and specialized ecological requirements showed elevated extinction rates, while generalists and those capable of reducing metabolic demands during environmental stress proved more likely to persist through the worst conditions. The End-Cretaceous extinction showed different selectivity, with body size playing a significant role as most land animals above approximately 25 kilograms in body mass went extinct, while smaller species showed higher survival rates, possibly reflecting differences in food requirements during the period of reduced primary productivity following the impact. These patterns are not perfect, and understanding why particular lineages survived while similar ones perished remains an active area of research involving new analytical techniques and continuing discovery of new fossil evidence. The megafauna extinctions of the late Quaternary period, occurring over roughly the past 50,000 years and continuing in accelerated form today, present a different category of extinction events with distinctive characteristics and causes that reflect the emergence of humans as a dominant force in global ecosystems. Unlike the mass extinctions of deep geological time, These extinctions affected primarily large-bodied terrestrial animals, showed strong temporal correlations with human arrival on different land masses, and occurred against a background of climate changes that complicated causal attribution. The pattern of these extinctions varies significantly across continents and islands in ways that shed light on the relative contributions of human activity and environmental change to megafauna disappearance. In Australia, the extinction of megafauna, including giant marsupials, large reptiles, and massive flightless birds, occurred approximately 45,000 years ago, following human arrival by perhaps a few thousand years. The precise timing remains debated due to dating uncertainties and limited fossil sites from the crucial period, but the coincidence of megafauna disappearance with human colonization, rather than with any identifiable climatic shift, has led many researchers to favour anthropogenic explanations. Species lost during this interval included Diprotodon, a herbivorous marsupial weighing perhaps 2,700 kilograms that represents the largest marsupial known from any time period, along with giant short-faced kangaroos, marsupial lions, enormous monitor lizards exceeding 5 meters in length, and the last surviving representatives of dramornithid birds, massive flightless herbivores that had dominated Australian ecosystems for millions of years. The Australian extinctions were particularly severe, eliminating perhaps 80 to 90% of megafauna species, a proportion that exceeds losses on other continents, and may reflect the particular vulnerability of animals that had evolved without hominin predators. The mechanisms by which human arrival precipitated megafauna extinction remain actively debated and likely varied across species and regions. Direct hunting, particularly of naive animals that had not evolved behavioural responses to human predators, certainly played a role in many cases, and archaeological evidence demonstrates that early human populations hunted various megafauna species. However, the magnitude of hunting required to drive continental-scale extinctions through direct killing alone would have been enormous, and some researchers emphasise indirect mechanisms including habitat modification through fire, disruption of keystone species interactions, introduction of diseases, and competition with other species influenced by human activity. The interplay between human impacts and climate change during the late Quaternary added further complexity, as both factors operated simultaneously and potentially synergistically, making it difficult to disentangle their relative contributions in specific cases. The woolly mammoth, perhaps the most iconic extinct animal of the Quaternary, exemplifies both the achievements and limitations of our ability to understand extinct species through diverse lines of evidence. Unlike most extinct animals, woolly mammoths are known not only from skeletal remains, but also from frozen carcasses preserved in permafrost regions of Siberia and Alaska, which have yielded information about soft tissue anatomy, hair structure and coloration, gut contents, parasites, and even intact cellular material suitable for genetic analysis. These exceptional specimens, combined with abundant skeletal and dental material from across the Northern Hemisphere, cave paintings and other artistic depictions created by humans who encountered living mammoths, and extensive trace evidence in the form of tracks, dung deposits, and modified vegetation make woolly mammoths among the best understood of all extinct species. The detailed knowledge available permits reconstructions of mammoth ecology, behaviour and physiology that would be impossible for species known only from fragmentary skeletal remains. Woolly mammoths possessed numerous adaptations to their cold environments, including thick coats of guard hairs reaching up to 90 centimeters in length, underlain by dense woolly underfur, reduced ears and tails that minimized heat loss from extremities, specialized tooth structures for processing abrasive steppe vegetation, and fatty deposits, including a distinctive neck hump that may have served for energy storage during harsh winter months. Genetic evidence reveals that mammoth hemoglobin had evolved modifications that improved oxygen delivery to tissues at low temperatures, a physiological adaptation that enhanced cold tolerance at the molecular level. Analysis of preserved gut contents and isotope signatures in teeth and bones indicates dietary flexibility that changed seasonally and geographically, with mammoths consuming grasses, sedges, flowering plants, shrubs, and occasionally tree material depending on its availability. These animals lived in herds with social structures probably resembling those of living elephants, as suggested by trackway evidence showing groups of varied sizes moving together and by the presence of extended family associations in death assemblages where multiple individuals died simultaneously. The extinction of woolly mammoths occurred not as a single event but as a gradual range contraction and population decline extending over thousands of years, with timing that varied significantly across different regions. Mammoths disappeared from most of their former range between approximately 15,000 and 10,000 years ago, during a period of rapid climate warming at the end of the last glacial period that transformed mammoth steppe habitat into forest, tundra, and other vegetation types less suitable for these large herbivores. However, isolated populations persisted much longer on islands that maintained suitable habitat, with mammoths surviving on St. Paul Island in the Bering Sea until approximately 5,600 years ago, and on Wrangel Island in the Arctic Ocean until approximately 4,000 years ago, well into the period when civilizations were developing in Egypt and Mesopotamia. These island populations experienced significant size reduction compared to mainland ancestors, a common phenomenon in island populations called insular dwarfism that reflects adaptation to reduced resource availability and relaxed predation pressure in island environments. The final extinction of the Wrangel Island mammoth population occurred under circumstances that remain unclear, but that likely reflected the accumulated consequences of small population size rather than any specific catastrophic event. Genetic evidence from Wrangel Island mammoths shows that this population experienced severe inbreeding, with individuals showing signs of harmful genetic mutations that accumulated as population size declined and genetic diversity eroded. These mutations affected various physiological functions including scent receptors, developmental processes, and coat appearance, potentially reducing individual fitness and population viability, even as environmental conditions remain stable. The Wrangel Island mammoths thus illustrate the importance of population genetic processes in extinction, demonstrating how small populations can enter extinction vortices in which genetic deterioration, demographic instability, and environmental fluctuations combine to drive further decline. The Americas experienced their own distinctive pattern of megafauna extinction coinciding with human arrival, though the timing was considerably later than in Australia, and the pattern of extinction showed both similarities and differences. Humans entered the Americas from Asia during the late Pleistocene, with the earliest well-documented sites dating to approximately 15,000 years ago, and the main colonization wave spreading rapidly across both continents over the following 2,000 to 3,000 years. Within this time frame, an extraordinary diversity of large mammals disappeared, including multiple species of mammoths and mastodons, ground sloths ranging from dog-sized forms to megatherium at weights exceeding 4,000 kilograms, glyptodonts with their massive armored shells, saber-toothed cats, American lions, dire wolves, short-faced bears, giant beavers, wild horses, camels, and numerous other species that had dominated American ecosystems for millions of years. The loss of this megafauna fundamentally transformed American ecosystems, eliminating keystone species that had shaped vegetation structure, nutrient cycling, and predator-prey relationships across entire continents. Ground sloths provide particularly instructive examples of extinct American megafauna because of their diversity, widespread distribution, and the quality of evidence available for understanding their ecology. More than 80 ground sloth species are known from the Americas, ranging across virtually all terrestrial habitats from Patagonia to Alaska, and including arboreal, semi-aquatic, and fully terrestrial forms, occupying diverse ecological roles. Unlike their living relatives, the tree sloths, which are small, specialized arboreal folivores, ground sloths exploited a wide range of food resources including leaves, fruits, roots, and in some cases animal material. The largest species, Megatherium americanum of South America, reached weights estimated between 4,000 and 6,000 kilograms and body lengths exceeding 6 meters when standing upright, making it among the largest land mammals ever to exist. This massive herbivore inhabited grasslands and open woodlands across much of South America until approximately 10,000 years ago, leaving evidence in the form of skeletal remains, skin and hair samples from dry cave deposits, dung balls containing identifiable plant material, and burrows excavated in sedimentary rock that can still be identified today. The preserved dung of ground sloths and other extinct herbivores has provided unexpectedly detailed information about the diets, behaviours and environments of these animals, allowing reconstruction of ecological relationships that would otherwise remain invisible. Shasta ground sloth dung from dry caves in the southwestern United States contains identifiable plant fragments, pollen, and other materials that reveal not only what these animals ate, but also what plants grew in their environments, and how they selected specific foods from available vegetation. Analysis of these deposits indicates that Shasta ground sloths consumed a variety of desert plants, including globe mallow, Mormon tea and yucca, digested their food thoroughly through extended gut retention times, and returned repeatedly to specific caves as shelter sites over periods of thousands of years. The dung deposits accumulated to substantial thicknesses in some caves, providing stratified records that can be dated and compared to track changes in diet and environment over time. Mummified remains of ground sloths recovered from caves in Patagonia and other arid regions have permitted direct observation of features that would otherwise require inference from skeletal anatomy. These specimens preserve skin with its characteristic pattern of bony ossicles embedded in the dermis, dense coats of reddish-brown hair, and in exceptional cases intact muscle tissue and internal organs. The quality of preservation in some specimens approaches that of recently deceased animals despite ages of more than 10,000 years, allowing detailed study of anatomy that would otherwise be entirely unknown. Early European settlers in Patagonia encountered these remains and sometimes interpreted them as evidence that giant sloths might still survive in remote areas, a possibility that motivated several expeditions into the interior during the late 19th and early 20th centuries, but that ultimately proved unfounded. The ecological consequences of megafauna extinction extend far beyond the direct loss of the species themselves to include cascading effects on vegetation communities, nutrient cycling, seed dispersal, and ecosystem structure that continue to shape American environments today. Many plants native to the Americas produce fruits or seeds that appear adapted for dispersal by large mammals that no longer exist, including avocados, Osage oranges, honey locusts, and various tropical fruits with large, nutritious fruits enclosing seeds that seem designed to pass through the digestive systems of animals far larger than any currently present. These anachronistic fruits suggest that the plants evolved in contexts that included mammoths, ground sloths, gomphotheres, and other mega herbivores as dispersal agents, and the absence of these animals may have altered plant distributions, reduced genetic exchange between populations, and contributed to vegetation changes that compound the direct effects of climate shifts during the Holocene. Some ecologists have proposed that the relatively sparse populations of large mammals in American ecosystems today, compared to equivalently productive environments in Africa, reflects not natural baseline conditions, but rather an impoverished state resulting from Pleistocene extinctions. Island extinctions present a distinct category of animal disappearance that differs in important ways from continental patterns and that provides particularly clear evidence of human causation due to the strong temporal correlations between human arrival and species loss on numerous islands worldwide. Islands support distinctive faunas with high proportions of endemic species found nowhere else, often including large-bodied representatives of groups that remain small on mainlands. Flightless birds evolved from flying ancestors, and species with reduced predator avoidance behaviours that reflect evolution in the absence of mammalian carnivores. These characteristics make island species particularly vulnerable to extinction following human arrival, when they encounter predators and competitors against which they have no evolved defences. The resulting pattern of island extinctions following human colonisation provides a natural experiment repeated dozens of times across the Pacific, Indian Ocean, Caribbean, and Mediterranean, consistently demonstrating the devastating effects of human arrival on naive island faunas. Madagascar illustrates these dynamics with particular clarity because of its large size, diverse endemic fauna, and relatively late human colonisation. Although humans reached Madagascar approximately 2,000 years ago, millennia after colonising most of Africa and thousands of years after reaching Australia, the island had until that point remained free of hominin influence, allowing its unique fauna to develop without the predation pressures that had already eliminated vulnerable species on other landmasses. Madagascar's extinct fauna included at least 17 species of lemurs larger than any living today, some exceeding 150 kilograms in body mass, along with elephant birds weighing up to 730 kilograms that represent the heaviest birds ever known, pygmy hippopotamuses, giant tortoises, giant fossas, and numerous other endemic species. The extinction of these animals following human arrival transformed Madagascar's ecosystems and eliminated species that had occupied key ecological roles as browsers, grazers, seed dispersers, and predators. Elephant birds deserve particular attention because of their extreme body size, distinctive ecology, and the detailed evidence available from diverse sources including skeletal remains, eggshell fragments, and accounts from early European visitors to Madagascar. These birds, comprising multiple species in the family Epiornithidae, were massive flightless herbivores that occupied ecological roles comparable to large ungulates on other landmasses, consuming fruits and vegetation while dispersing seeds through their digestive systems. The largest species, Vorombi titan, reached heights of approximately 3 meters and masses estimated around 730 kilograms, making it the heaviest bird known from any time period. Elephant bird eggs, which measured up to 34 centimeters in length and held volumes of approximately 9 liters, are commonly found as fragments in archaeological sites and occasionally recovered intact, suggesting that egg collection by humans may have contributed to population declines. Radiocarbon dating of elephant bird remains indicates that at least some species survived until approximately 1,000 years ago, meaning that they overlapped with human occupation for roughly a millennium before extinction. The giant lemurs of Madagascar present similar patterns of survival after human arrival, followed by extinction within the past 1,000 years, though the mechanisms of their disappearance may have involved combinations of hunting, habitat destruction, and climate change that varied across species and regions. Unlike the smaller lemurs that survived to the present, the giant lemurs occupied ecological niches that may have made them particularly vulnerable to human impacts, including slow reproduction, specialized diets, and behaviors that brought them into frequent contact with humans. Species such as Archaeoindris, a ground-dwelling lemur roughly the size of a male gorilla, and Paleopropithecus, a sloth-like lemur that hung from branches using highly modified limbs, represented adaptations to specific ecological conditions that left them with limited ability to adjust to rapid environmental changes. Subfossil sites in Madagascar sometimes contain remains of giant lemurs associated with evidence of butchery marks or burning, indicating that at least some populations were hunted, though the relative importance of direct hunting versus habitat modification remains debated. New Zealand provides another instructive case of island extinction following human colonization, with the advantage that the chronology is exceptionally well established through radiocarbon dating, and the period of extinction is comparatively recent and brief. Polynesians reached New Zealand approximately 700 to 800 years ago, encountering an avifauna dominated by birds that had evolved in the complete absence of mammalian predators, and that included numerous flightless species uniquely vulnerable to hunting. The emua, comprising 9 species of large to enormous flightless birds in 2 families, represent the most famous casualties of this colonization, but extinctions extended to many other species, including Haast's eagle, the largest eagle known with a wingspan exceeding 3 meters and body mass around 15 kilograms, several species of waterfowl, rails, and songbirds, along with various reptile and invertebrate species. The extinction of this fauna occurred extremely rapidly, with most species disappearing within approximately 100 to 200 years of human arrival, far too quickly to attribute to climate change, and clearly linked to intensive hunting and habitat modification through fire. Haast's eagle deserves attention not only for its impressive size, but also for the ecological role it occupied as the apex predator in New Zealand ecosystems, and for what its extinction reveals about the interconnectedness of species within ecological communities. This enormous raptor evolved from a much smaller ancestor that reached New Zealand, and subsequently increased in size to exploit the large-bodied moa as prey, representing an example of island gigantism in a predator corresponding to the gigantism of its herbivorous prey. Analysis of skeletal proportions indicates that Haast's eagles combined massive talons, capable of puncturing bone with relatively short wings, adapted for maneuvering in forested habitats, suggesting that they attacked moa through surprise ambush rather than the soaring pursuit typical of other large eagles. The extinction of moa eliminated the prey base that supported Haast's eagle populations, demonstrating how species extinctions can cascade through food webs to affect predators, competitors, and mutualists of the directly eliminated species. The dodo of Mauritius occupies a unique position in public consciousness as perhaps the most widely recognized symbol of human-caused extinction, though many commonly held beliefs about this bird rest on limited evidence or historical misunderstandings. The dodo was a large flightless pigeon weighing approximately 10 to 18 kilograms that evolved in isolation on Mauritius from flying ancestors that reached the island millions of years ago. Like many island species, the dodo had lost both the ability and the instinct to flee from predators, making it extremely vulnerable to the sailors, colonists, and introduced animals that arrived with Dutch settlement beginning in 1598. The last confirmed dodo sightings date to the 1660s, giving the species a known overlap with humans of less than 80 years and making its extinction among the most rapid documented for any species. The brevity of the period during which dodos were observed by literate Europeans means that accounts of their behaviour, ecology, and appearance are limited and sometimes contradictory. Popular depictions of the dodo as obese, clumsy, and almost comically maladapted reflect not accurate biological understanding, but rather the combination of historical caricature, specimens that may have been overfed in captivity, and the general tendency to emphasize the strangeness of extinct species. Analysis of dodo skeletal remains indicates that these were robust but not particularly fat birds, well adapted to their forest floor niche and capable of moving efficiently through their native habitat. The eggs and chicks of dodos, laid on the ground and unable to escape predators, would have been exceptionally vulnerable to the rats, pigs and monkeys that Europeans introduced to Mauritius, and this reproductive vulnerability may have contributed as much to extinction as direct hunting of adult birds. The dodo's fame as an extinction symbol reflects historical accidents of timing and documentation, more than any intrinsic characteristics of the species, but this fame has given the dodo an enduring role in discussions of biodiversity loss and conservation. Moving from islands to continental extinctions of the historical period reveals different patterns and mechanisms that reflect the distinctive ways humans have transformed mainland environments and exploited mainland wildlife. Continental species generally occupied larger ranges, maintained larger populations, and coexisted with human hunters for longer periods than island species, providing them with greater resilience to hunting pressure, but exposing them to accumulating impacts from habitat conversion, introduced species, and technological intensification of exploitation. The extinctions of species such as the passenger pigeon, Carolina parakeet and thylacine occurred not through the naive vulnerability of island species, but through industrial-scale exploitation, habitat destruction, and persecution that exceeded the reproductive capacity of even enormously abundant species. The passenger pigeon represents perhaps the most dramatic example of how rapidly a superabundant species can be driven to extinction when exploitation pressures become sufficiently intense and habitat destruction eliminates the conditions necessary for successful reproduction. Historical accounts indicate that passenger pigeons numbered in the billions during their peak abundance, forming flocks that darkened the sky for hours as they passed, and nesting colonies that covered hundreds of square kilometers of forest. Observers described flocks a mile wide passing overhead continuously for hours, blocking out sunlight and producing noise audible at great distances, suggesting population densities that seem almost inconceivable from a modern perspective. The last passenger pigeon, a captive female named Martha, died at the Cincinnati Zoo on September 1, 1914, completing an extinction trajectory that had accelerated over the preceding decades through the combined effects of commercial hunting and deforestation. The ecology of passenger pigeons reveals why this species proved unexpectedly vulnerable to extinction, despite its apparently overwhelming numbers. Passenger pigeons depended on mast crops, the intermittent heavy production of nuts and seeds by trees such as oaks, beeches, and chestnuts that occurred in different locations in different years, requiring the birds to range widely across eastern North America to locate sufficient food sources. The massive flocks represented an adaptation to this unpredictable resource distribution, allowing the birds to locate and exploit mast crops efficiently through collective searching and information sharing. Reproduction also occurred in enormous colonies that may have depended on reaching critical population densities to trigger breeding behaviour and to swamp predators through sheer numbers, making the bird vulnerable to disruption of colonial nesting through hunting and disturbance. As commercial hunting reduced populations and deforestation eliminated nesting habitat and food sources, the species entered a decline spiral in which reduced numbers interfered with the social behaviours necessary for successful reproduction, accelerating population decline beyond what hunting and habitat loss alone would have caused. The Carolina parakeet, the only parrot species native to the eastern United States, disappeared through a similar combination of factors that illustrate how multiple pressures can combine to eliminate species that were once widespread and locally abundant. Historical accounts place Carolina parakeets in flocks of hundreds of individuals across the southeastern United States, where they fed on seeds of various plants, including agricultural crops that brought them into conflict with farmers. The birds possessed a fatal tendency to return to locations where flock members had been killed, hovering over the bodies and allowing hunters to shoot additional individuals, a behaviour that made hunting exceptionally efficient and that may have reflected social bonds that proved maladaptive under sustained persecution. Habitat destruction eliminated the old-growth bottomland forests that provided nesting sites and food sources, while hunting, capture for the pet trade, and possibly disease contributed to population declines. The last confirmed wild Carolina parakeet sightings date to the early 1900s, and the last captive individual died in 1918, just 4 years after the last passenger pigeon. The thylacine or Tasmanian tiger provides a case study in extinction that spans both prehistoric and historical periods, and that illustrates how human impacts can eliminate species across multiple timeframes and through multiple mechanisms. Thylacines were marsupial carnivores that superficially resembled dogs or wolves, but represented an independent evolutionary lineage that had diverged from other marsupials tens of millions of years ago, demonstrating through their morphology the phenomenon of convergent evolution, in which unrelated lineages independently evolve similar features in response to similar ecological pressures. Thylacines once occurred across mainland Australia and New Guinea, in addition to Tasmania, but they disappeared from these larger land masses approximately 3,000 years ago, likely due to competition with dingoes and possibly intensified human hunting. The Tasmanian population survived because Tasmania had separated from the Australian mainland before dingo arrival, providing a refuge free from this competitor. European settlement of Tasmania beginning in 1803 initiated a rapid decline that would lead to thylacine extinction within approximately 130 years. Settlers viewed thylacines as threats to livestock, particularly sheep, and the Tasmanian government instituted bounty programs that paid for dead thylacines, directly subsidising the killing of approximately 2,000 individuals between 1888 and 1909. This persecution, combined with habitat destruction, reduced prey availability and possibly diseases transmitted by domestic dogs, eliminated thylacines from most of Tasmania by the early 20th century. The last known thylacine died at the Hobart Zoo on September 7, 1936, and searches since that time have failed to confirm the continued existence of the species, despite occasional unverified sightings and persistent public interest in possible survival. Historical film footage and photographs of thylacines, while limited, provide visual documentation of this extinct species that is available for very few other animals lost in the historical period. The footage shows a large, long-bodied marsupial with distinctive striped hindquarters, moving with a somewhat stiff gait that reflected anatomical characteristics, differentiating it from the placental carnivores it superficially resembled. These images, combined with preserved specimens in museums and detailed descriptions from observers who encountered living thylacines, permit unusually confident reconstructions of this species' appearance and behaviour. Nevertheless, many aspects of thylacine ecology and behaviour remain uncertain, including the details of their hunting strategies, social organization, reproductive behavior, and vocal communication, because sustained scientific study was not conducted while the species remained sufficiently abundant for such research. The great auk represents the original penguin, a large flightless seabird of the North Atlantic that was driven to extinction through hunting for food, feathers, and oil during the centuries of European maritime expansion. Great auks bred on rocky islands in the North Atlantic, where their inability to fly and their colonial nesting habits made them exceptionally vulnerable to human exploitation. Sailors visiting these colonies took eggs, chicks, and adults for food, while later exploitation focused on the valuable down feathers used in bedding and the oil that could be rendered from their fat. As populations declined and great auks became rare, specimen collectors intensified their efforts to obtain skins and eggs before the species disappeared entirely, ironically contributing to the very extinction they sought to document. The last confirmed great auks were killed on Eldey Island off Iceland on June 3, 1844, with the final specimens strangled by collectors who subsequently sold the skins and eggs to natural history museums. The Steller's sea cow, a massive marine mammal related to manatees and dugongs, experienced one of the fastest documented extinctions of any large animal, disappearing within approximately 27 years of its discovery by Europeans. German naturalist Georg Wilhelm Steller encountered and described this species in 1741 during the ill-fated Bering expedition, which was shipwrecked in the Commander Islands of the Western Bering Sea. Steller documented a marine mammal reaching lengths of 8 to 9 meters and weights of possibly 8,000 to 10,000 kilograms, making it among the largest marine mammals and the largest member of the order Sirenia known from any time period. Steller's sea cows inhabited the kelp beds surrounding the Commander Islands, and possibly formerly occurred more widely across the North Pacific, before human hunting reduced their range. The stranded expedition members survived partly by hunting sea cows for food, and subsequent visitors to the islands continued this practice, until the species was extinct by approximately 1768. Steller's observations provide the only detailed account of sea cow behavior and ecology, describing animals that were apparently tame and unafraid of humans, that lived in groups showing social bonds, that fed continuously on kelp in shallow waters, and that apparently mated during the spring months. The thick skin and abundant fat of sea cows made them valuable to hunters, as a single animal could provide meat for an entire crew for an extended period, and the fat rendered into oil suitable for lamps and cooking. The small population inhabiting the Commander Islands, possibly numbering only 1,000 to 2,000 individuals at the time of the European contact, had no capacity to sustain even modest hunting pressure, and the combination of food hunting, oil production, and incidental killing during seal and sea otter hunting drove the species to extinction within a human generation of its discovery. The quagga, a distinctive form of plain zebra with reduced striping on its hindquarters and legs, disappeared from its native range in the southern Africa during the 19th century through a combination of hunting and habitat competition with livestock. Whether the quagga represented a full species, a subspecies of the plain zebra, or merely a regional population with distinctive coloration has been debated, but genetic analysis of preserved specimens has confirmed that quaggas were genetically nearly identical to living plain zebras, and that their distinctive appearance likely reflected a localized color variant, rather than deep evolutionary separation. The last wild quaggas were probably killed in the 1870s, and the last known individual, a mare in the Amsterdam Zoo, died on August 12, 1883. Because quaggas were closely related to living zebras, a project called the Quagga Project has attempted to recreate animals with quagga-like coloration through selective breeding of plain zebras with reduced striping, though these animals, whatever their appearance, cannot represent a genuine restoration of the extinct population and its genetic lineage. The baiji, or Chinese river dolphin, represents the first known extinction of a cetacean species in modern times, disappearing from the Yangtze River by the early 2000s, despite decades of conservation concern and some protective measures. Baiji were freshwater dolphins that had inhabited the Yangtze River system for millions of years, evolving numerous adaptations to life in turbid, murky water, including very poor eyesight, compensated by sophisticated echolocation abilities. The species survived through most of the 20th century, despite declining water quality and increasing human activity on the river, but populations declined precipitously during the decades of rapid economic development in China, reaching critically low levels by the 1990s. A comprehensive survey in 2006 failed to detect any surviving individuals, and the species is now considered functionally extinct, representing the loss of a unique evolutionary lineage that had diverged from other dolphins approximately 20 million years ago. The causes of baiji extinction included multiple human impacts that interacted in ways that made the river increasingly inhospitable to dolphins. Bycatch and fishing gear, particularly rolling hook long lines and electric fishing equipment, directly killed many individuals. Vessel strikes from the heavy commercial traffic on the Yangtze injured and killed dolphins attempting to navigate through shipping lanes. Pollution from agricultural runoff, industrial discharge, and municipal waste degraded water quality and may have affected preyfish populations. Construction of dams, including the massive Three Gorges Dam, altered river hydrology and fragmented habitat in ways that disrupted dolphin movements and breeding. The combination of these factors exceeded the capacity of the small remaining population to reproduce faster than individuals were lost, ultimately driving the species to extinction despite recognition of its endangered status and some attempts at conservation intervention. The gastric brooding frogs of Australia, comprising 2 species discovered only in the 1970s and 1980s, exemplified how recently discovered species can disappear before science has opportunity to fully understand them. These remarkable frogs possessed a unique reproductive strategy in which females swallowed their fertilized eggs, suspended gastric digestion through production of specialized chemicals, and brooded their developing offspring in their stomachs for 6 to 8 weeks before regurgitating fully formed froglets. This reproductive mode, unknown in any other vertebrate, held potential interest for medical research into control of gastric secretions, but was lost before detailed study could be completed. Both species disappeared during the 1980s, likely through the effects of the chytrid fungus Batrachochytrium dendrobatidis that has driven numerous amphibian extinctions worldwide. Combined possibly with drought conditions and other environmental stresses, the role of disease in animal extinctions has received increasing attention as the impacts of pathogens on wild populations have become better understood, revealing disease as both a proximate extinction mechanism and an underlying factor that can interact with other stressors to drive population collapse. Introduced diseases carried by domestic animals, humans, or other introduced species have contributed to numerous extinctions, from the potential role of avian malaria in Hawaiian honeycreeper, declines to the suspected contribution of disease to thylacine extinction. The chytrid fungus affecting amphibians demonstrates how a novel pathogen can spread globally and cause population crashes and extinctions across multiple continents, affecting species that had persisted through previous environmental changes and human impacts. Understanding disease dynamics in wildlife populations has become essential for predicting and preventing extinctions, though the cryptic nature of many pathogens and the difficulty of studying disease in wild populations limit what can be known about disease impacts on historical and prehistoric extinctions. Climate change has interacted with other extinction drivers throughout Earth history, and continues to function as both direct and indirect cause of species decline and disappearance in the contemporary period. Past climate changes, including the dramatic shifts at the ends of ice ages, the warm intervals of the Eocene, and the glacial-interglacial cycles of the Quaternary, have driven extinctions, range shifts, and evolutionary changes across diverse taxa and regions. However, species have also demonstrated remarkable resilience to climate changes that occurred gradually enough to allow range shifts, adaptation, and adjustment of ecological relationships. The current period of anthropogenic climate change differs from many past episodes in its rapidity, its combination with habitat fragmentation that impedes range shifts, and its occurrence against a background of already depleted populations and degraded ecosystems that reduce the resilience species can bring to responding to changing conditions. The fossil record provides context for understanding how species have responded to past climate changes, revealing both extinctions and survivals that illuminate the factors determining species vulnerability. during the end Pleistocene warming that accompanied the transition from glacial to interglacial conditions, many cold-adapted species experienced range contractions or extinction as their preferred habitats disappeared, while other species expanded their ranges northward to track suitable conditions. The woolly rhinoceros, a cold-adapted species that coexisted with woolly mammoths across much of northern Eurasia, disappeared approximately 14,000 years ago during a period of rapid warming, though whether climate change alone was responsible or human hunting contributed remains debated. In contrast, species such as the musk ox and Arctic fox successfully tracked suitable habitat to high Arctic regions where they persist today, demonstrating that climate-driven range shifts could provide alternatives to extinction for species with sufficient mobility and adaptability. The importance of refugia, isolated areas where suitable conditions persisted despite regional or continental climate changes, appears repeatedly in extinction and survival patterns. Island populations of various species, including the Wrangel Island mammoths discussed earlier, survived for thousands of years after mainland populations disappeared, suggesting that suitable microhabitats can provide persistence opportunities that large-scale climate reconstructions might not predict. Mountain regions have similarly served as refugia, where species could persist by shifting altitudinally in response to temperature changes, tracking suitable conditions up slopes during warming and down during cooling. The existence and accessibility of refugia depends on geographical circumstances that vary among species and regions, helping to explain why similar climate changes produce different extinction patterns in different locations. The ecological roles occupied by extinct animals represent permanent losses to Earth's biodiversity that extend beyond the species themselves to encompass unique evolutionary solutions, ecological functions, and potential future adaptations that disappear with each extinction. Large herbivores such as mammoths, ground sloths, and various proboscideans shaped vegetation communities through their feeding, trampling, and seed dispersal, maintaining landscapes in states that disappeared when these animals were lost. Apex predators such as saber-toothed cats, thylacines, and Haast's eagles regulated prey populations and competitor species in ways that restructured entire ecological communities following their removal. Specialized pollinators, seed dispersers, and mutualists co-evolved with partner species that may persist, but with altered ecology in the absence of their former associates. Attempts to understand and potentially restore extinct ecological functions have motivated various initiatives ranging from rewilding with ecological proxies to speculative proposals for de-extinction using genetic technologies. Rewilding approaches that introduce living species as functional replacements for extinct ones have been implemented in various contexts, from the reintroduction of large herbivores to European landscapes to proposals for introducing elephants to North American grasslands as mammoth proxies. These approaches raise complex ecological, ethical, and practical questions about whether introduced species can genuinely replicate the functions of extinct ones, and about the consequences of introducing species to ecosystems from which they have been absent for thousands of years. De-extinction proposals, which envision using preserved genetic material to recreate extinct species, or engineering close relatives to express extinct species traits, face even more fundamental questions about technical feasibility, ecological relevance, and ethical appropriateness. The temporal distribution of extinctions reveals patterns that reflect the progressive expansion of human impact across the planet and the intensification of human pressures on remaining biodiversity. The earliest documented human-caused extinctions occurred in Australia approximately 45,000 years ago, followed by extinctions on Pacific Islands as Polynesian navigators colonized archipelagos across the ocean, then by extinctions in the Americas following human arrival, and subsequently by extinctions on remaining oceanic islands as European expansion reached Madagascar, the Mascarene Islands, New Zealand, and other previously uncontacted lands. Continental extinctions accelerated during the industrial period as new technologies enabled more intensive exploitation of wildlife and more rapid transformation of habitats. The current period of accelerated extinction, sometimes characterized as a sixth mass extinction, represents the continuation and intensification of processes that have operated throughout the Anthropocene the proposed geological epoch defined by human dominance of Earth systems. Understanding extinct animals requires engaging not only with biological questions about species ecology, evolution, and extinction mechanisms, but also with broader questions about the nature of scientific knowledge and the interpretation of inherently incomplete evidence. Every reconstruction of an extinct species involves inferences that bridge gaps between available evidence and conclusions about appearance, behavior, ecology, and evolutionary relationships. These inferences draw on comparative anatomy with living relatives, functional analysis of preserved structures, ecological theory about niche occupancy and community structure, and various forms of chemical, isotopic, and genetic analysis that extract information from preserved tissues. The resulting understanding is provisional, subject to revision as new specimens are discovered and new analytical techniques become available, and necessarily incomplete in ways that cannot be fully specified. The discovery of soft tissue preservation in some dinosaur fossils illustrates how unexpected findings can transform understanding of groups previously known only from skeletal remains. While most dinosaur fossils preserve only bone, exceptional specimens from sites with unusual preservation conditions have revealed feathers, skin impressions, gut contents, and possibly even pigment molecules that enable colour reconstruction. These discoveries have revolutionized understanding of dinosaur appearance and behavior, revealing that many theropod dinosaurs were feathered, that colour patterns could be complex and possibly used in display and camouflage, and that various species preserved evidence of specific meals, diseases, and injuries in their last days of life. Similar advances in analytical techniques continue to extract new information from existing specimens, as methods for analysing ancient proteins, isotopes, and genetic material become more sensitive and applicable to older and more degraded specimens. The development of techniques for recovering ancient DNA has opened entirely new categories of investigation into extinct species, enabling direct analysis of genetic relationships, population history, and in some cases, physiological adaptations that would be invisible in morphological evidence alone. Ancient DNA has clarified the evolutionary relationships among mammoths, elephants, and mastodons, confirmed the close genetic similarity between quaggas and plain zebras, revealed that Neanderthals interbred with anatomically modern humans, and provided insight into the inbreeding depression that affected the final mammoth populations on Wrangel Island. These analyses are limited by the degradation of DNA over time and by preservation conditions that favour cold, dry environments over tropical settings, but they continue to expand in scope as extraction and sequencing technologies improve. The recovery of genetic material from extinct species has also raised the speculative possibility of de-extinction, the recreation of extinct species through genetic engineering of preserved DNA, or through modification of related living species to express extinct traits. Woolly mammoths have been prominent candidates for such efforts because of the availability of the well-preserved genetic material from frozen specimens and the existence of close living relatives in elephants that could potentially serve as surrogate parents for reconstructed embryos. However, the technical, ecological, and ethical challenges involved in such projects remain formidable, including the complexity of reconstructing complete functional genomes from degraded fragments, the difficulty of producing viable embryos with modified genomes, the question of what habitat and social environment would be appropriate for individuals of resurrected species, and broader ethical questions about the appropriateness of such interventions. The lessons available from extinct animals extend beyond the specific circumstances of individual species disappearances to encompass general insights about the conditions that create vulnerability to extinction and the mechanisms that drive population decline to the point of irreversibility. Small populations face heightened extinction risk from both deterministic factors, such as continued exploitation or habitat loss, and stochastic factors such as disease outbreaks, natural disasters, demographic fluctuations, and genetic problems associated with inbreeding. Species with specialized habitat requirements, narrow geographical ranges, slow reproduction, and limited behavioral flexibility show elevated vulnerability across diverse extinction episodes. The interaction of multiple threats often proves more dangerous than any single factor, as the combined effects of hunting and habitat loss, climate change and disease, or exploitation and competition can exceed the adaptive capacity of species that might survive any of these stresses individually. The contemporary period of biodiversity decline represents a continuation of the extinction processes that have characterized the past 50,000 years of human expansion, but at accelerated rates and intensified scales that raise unprecedented conservation challenges. Current extinction rates are estimated to be 100 to 1,000 times higher than background rates in the fossil record, with continued acceleration projected as climate change, habitat destruction, pollution, invasive species, and direct exploitation combine to affect remaining populations. Species currently on trajectories toward extinction include representatives of most major vertebrate groups and many invertebrate and plant lineages, though the extent of contemporary extinction in less studied groups such as insects and marine invertebrates remains poorly characterized. The study of extinct animals ultimately serves as both a scientific discipline, investigating specific biological questions, and a source of perspective on the current state and possible futures of Earth's biodiversity. Understanding why species went extinct, how they lived before their extinction, and what their losses meant for the ecosystems they inhabited provides context for evaluating contemporary conservation challenges and the effectiveness of proposed interventions. The extinct species discussed here represent a tiny fraction of the animals that have disappeared throughout Earth history, and even a small sample of those lost during the period of human influence. But their diversity illustrates the range of circumstances, mechanisms, and consequences that characterize extinction as a biological and historical process. The irrecoverability of extinction makes each species' disappearance a permanent subtraction from the inventory of biological diversity that evolution has generated over billions of years. Unlike habitat destruction, which can sometimes be reversed, or population decline, which can potentially be addressed through conservation intervention, extinction is an absolute boundary beyond which no recovery is possible, at least with current technology. This finality distinguishes extinction from other forms of environmental degradation and gives special weight to decisions that affect the survival prospects of endangered species. The extinct animals of the geological and historical past cannot be restored, but understanding their stories contributes to the knowledge base that informs efforts to prevent further losses among the species that remain.